Hi, my name is Alexandra, and I'm here to share with you my project on Zwitterionic-nanoparticle dressing targeting inflammation and reactive oxygen species in diabetic wounds. My mentors include Dr. Matthew Osmond, Michael Stager, and Dr. Melissa Krebs. We chose to target diabetic wounds due to the vast impact that they have on these diabetic patients. It takes a lot of time and money to get these wounds to heal properly, and when they are not properly healed, there's a possibility of losing a limb. We hope to give these patients a more efficient and less costly means of drug delivery with our Zwitterionic gels. We chose Zwitterionic hydrogels because they have shown to have a self-healing ability, and so even if these gels were to split apart, it would be able to heal itself and form a single gel again. They have also shown the capability of being able to be injected, which would be extremely helpful because then the gel itself could be injected straight into the wound and cross-linked using a light to fit the entire wound, and every part of the surface of the wound would get a sufficient drug delivery. Each gel has a mixture of two monomers. One is HEMA and the other is either this CBMA or the SBMA monomer. And basically the only difference between these two monomers is this sulfon group right here. And the reason I would choose one over the other during my experiments was mainly availability. Our supplier stopped making the CBMA, so during the time when we were developing our own synthesis for the CBMA, I was mostly relying on the SBMA, but after we successfully were able to make the CBMA, I just kind of chose between the two based on the experiment. And then the zwitterionic component of both of these monomers gives an extra interactive force between the polymers and helps give them their gelatinous and self-healing state, which could be seen on the last slide. We also use a cross-linking agent, PEGMA, which gives these gels even more stability and the ability to hold their shape. The photo initiator that we use, which is a combination of Ruby and SBS, gives us control over where and when we want the gels to polymerize. So basically we can have the gel inserted right into the wound and then polymerize it right there using a photo initiator. And then to determine the best procedure for making these gels, a variety of tests for the polymers, monomer to monomer, and cross-linker concentrations were performed. Based off these tests, we were able to determine a good range of concentrations that gave us the desirable characteristics. As you can see on this left figure, I was using a 20% polymer solution, and then also I was varying my PEGMA concentrations and then also my SBMA monomer to HEMA. And then you can kind of see that these are more of the gels we want because these ones with the higher concentration of PEG were too brittle and stiff, and kind of in this last one you could see they would break apart and then they wouldn't form a coherent gel again. And then we also did a swelling test on these gels to see how they would react in an aqueous solution. And as you can see, many of the lower concentrations kind of kept their shape. They broke apart a little bit, but the 10% definitely kept their shape. And then as we got higher concentrations of SBMA, they swole a lot more. And then from this, we could kind of determine that we wanted to stay around the range of the 0.1% PEG, and then also lower concentrations of SBMA, so anywhere from 10 to 25%. And then, so the next step for this project would be a small molecule release, and then also cell studies. We need to make sure that when we insert the drug into the gel, it's a slow release of the gel or of the drug from the gel, so that it's not all just releasing the drug all at once, because we want the wound to be receiving the beneficial drug factors for a prolonged period of time instead of just one application. And then also the cell studies would provide us with information of whether it's biocompatible and if like cells would be able to grow around the gel and help heal the wound. And then these gels, we believe, provide a huge breakthrough in the treatment of chronic wounds, and then also provide a better option for treatment. Since it would be a one-time application, you would not have to be re-exposing the wound to the environment multiple times, like the application or the treatment options that are available today. And then we just want to give a huge thank you to the Colorado School of Mines, the Children's Hospital of Colorado, and the Colorado Office of Economic Development and International Trade for providing us funding on this problem, or on this project. Thank you for your time.